Hi, everyone. Thank you for joining. This is Sabrina Paganoni. I'm speaking from the HEALEY and AMG Center for ALS at Mass General Hospital in Boston. And we're giving everyone a few seconds to join us from the waiting room. Thank you for your patience. And we can go to the next slide. And today is a special webinar for the platform trial webinar series. As you know, we are trying to schedule content and meetings around the questions that matter the most to the community. Many people have asked us to explain the drug development process in greater detail, and obviously the same principles also apply to the platform trial and other trials that are, again, part of the drug development process as a whole. And so tonight, to help answer some of the questions that we've been getting from many of you, if you can go to the next slide, we continue our webinar series. This is a list of our upcoming discussions, but for tonight, next slide, we are really excited to talk about, again, ALS drug development, the basic principles, the main principles that govern this process. And we're really excited to have two experts on the line today. So we have Dr. Su from Penn State, and I'll let Dr. Su introduce himself. He's an investigator with great experience in ALS clinical research. Will tell us more about the main approach in ALS drug development, and then we'll have Dr. Robinson after him. So without further ado, I'll give it up to you, Dr. Su.
Okay, and it's my pleasure to be here. I'm Bill Su at the Penn State Hershey Medical Center. I'll give the first of the two parts of this webinar on drug development. And next slide. And so my first slide is the busiest. I really want to use this as kind of a visual representation of some of the frustrations and the challenges we've faced in the whole process over the past few decades. And so this slide highlights that over the years we've come to understand more and more about various different pathological processes in the diseased motor neurons in individuals who have ALS. And we've had increasing numbers of potential drug candidates targeting each of these processes. However, despite all of that work, at this point, we have a frustratingly few number of compounds that are approved for use in humans. So in the United States, we have Riazole, as well as edaravone or Radicava, and then tofersen, which is approved only for SOD1 familial ALS. And in Europe, we just have Riazole. And so this part of the talk really tries to address what are the issues in both the preclinical or the before human stage, as well as human clinical trials that can be targeted to try to improve the process. So next slide. So overall, for any kind of drug development, we have two major phases. The first part, we look at different non-human organisms to do one of kind of broadly two categories. We can take different types of organisms as models, and that's why we call these model systems, as models for the disease processes themselves. So we can take simpler organisms or particularly important organisms to model different parts of, for example, motor neuron degeneration. Or we can take a non-human system and try to see if the drugs, the compounds that we've identified may have some activity in a non-human model of ALS. And so I'll highlight two of these as examples, prototypical examples. If you click for the animation. So we have now the ability to take human cells. So these might be skin cells or other accessible cells from individuals with ALS, whether familial or sporadic, and then reprogram them using stem cell type technologies to become a more pluripotent cell and then program them back into a motor neuron. This is a very powerful model where we basically have a cell that is a human cell with the same genetics as someone who had disease. And obviously, we'd be able to study the pathophysiologic processes in a controlled environment. Next animation. We also have several different mouse models of disease. The classic one, the SOD1 mouse model, has a very well-defined progression of symptoms that somewhat mimic the progression in humans. The goal here would then be to take the most promising candidates and apply it to the system to see if it has a similar impact in the mouse model. So, for example, Rizal has an impact. Tofersen had an impact in the SOD1 mouse model as well. So the goal in this preclinical stage, we have many, many different compounds, but we only want to select the most promising ones to use in humans, promising in terms of potential effectiveness as well as safety. Next slide. The problem then becomes, there's several challenges at the preclinical stage that is a bottleneck for drug discovery. I'll highlight three of these. Any of the types of model systems that we have available, unfortunately, don't fully replicate what we can see in an individual with ALS. So, for example, in the cell line model I talked about, even though these are true motor neurons, in a cell culture, you have at most just a few different types of cells. It lacks all of the different central nervous system structures and connectivity, and it doesn't fully model the disease process, certainly not longitudinally over the decades that someone might live. In the mouse model, sure, we have the ability to look at the impact of drugs over the lifespan of an animal that mimics a human disease, but that lifespan is on the order of 3 to 5 months to really go through a full experiment. And that's just one experiment that might need replication. So it's very difficult to do this in a high throughput fashion. For next animation. So once you identify the targets that you really want to target in a human, the next problem becomes that because the model systems themselves are what we're working with, the drugs that we identify may not affect the human central nervous system receptors or structures or proteins the same way. We have to go through several different iterations of a drug candidate before we really are able to ensure that it targets the way or the human that we want it to. And final animation on this slide. And lastly, even in the non-human animal systems, finding the correct time points to test the drugs is somewhat problematic, right? So in the mouse model, many of the drugs that initially were promising were actually started well before a symptomatic stage. So, this is not something that we could do in a sporadic person with ALS, right? We can't give everybody who may develop ALS a drug because we have no idea who may or may not develop ALS. Next slide. Nonetheless, we have taken many, many different compounds from the preclinical stage into the clinical trial phase. And I'll go over the different phases of clinical trials very briefly. This is taken from data from the FDA for clinical trials in general. So the numbers here, the time points aren't specific for ALS. Phase 1 trials, the basic principle, the goal is to determine, are the compounds safe to use in humans? Typically, these enroll healthy participants, healthy volunteers. The numbers are slightly smaller, from 20 to 100 on average for all comers, all diseases. And the length is usually somewhat short, up to 6 months. A majority of trials actually pass phase 1. So by the time they reach clinical trials, these are mostly safe. 70% will pass. Once a compound enters phase 2, the goal is, 1, to determine, is the drug safe in people with that disease? And 2, is there some signal for efficacy that we want to continue? These trials are about a year to a year and a half, anywhere from, on average, 100 to 300 participants. And on average, about a third of drugs that enter phase 2 will proceed to phase 3, 33%. Phase 3 trials are the definitive, large, double-blind, placebo-controlled trials that lead to FDA approval if successful. The goal here really is to determine, is the drug effective? Are there specific risks, benefits in subpopulations? Are biomarkers affected? These enroll up to 3,000 participants or more, depending on the disease. They might last up to 2 to 3 years or even longer. And on average, about a quarter of compounds that enter phase 3 show some sign of efficacy and are approved. Next slide. So let's compare that with ALS. So again, in summary, if you take the FDA data on average and you have 100 drugs that enter phase 1, that enter the earliest human testing, only about 6 of them will pass phase 3 and be deemed effective. So, right, that's 70% from phase 1 to phase 2, 33% into phase 3, and then 25% of phase 3. Next animation. I found this pretty, or actually a very excellent review of the results of ALS trials over a 12-year period from 2008 to 2019. In that paper, they identified approximately 100 phase 2 trials or beyond. In this study, they saw that the percentage of trials that passed phase 2 was somewhat comparable to the overall trial experience, right? 20% versus 33%. However, only one of these, or 5%, that passed phase 2 passed phase 3. And in this case was edaravone radicava. Next animation. And so 4 ALS drugs have really been approved by the FDA for use. Riazole occurred in the mid-1990s, and that was a very large phase 3 trial, almost 1,000 participants. The other 3 compounds listed here had somewhat smaller enrollment. Next slide. So, why is that, and what can we do, and what's being done to optimize trials themselves to meet the needs of people with ALS? We recognize now that ALS itself is not like your typical disease. A trial for ALS is not like a trial for high blood pressure or even diabetes, something like that, right? ALS is a more severe disease. It is rare, and there is a clear need for better treatments. And so we have to balance large, long studies that allow us to gather more statistically powered information with equity and justice. We don't want to expose too many people with ALS, placebo. We want to have as many people in the clinical trial have the chance of being exposed to the active compound, given their participation and their willingness to undergo the side effects of that potential drug. And so some of the trials, for example, the HEALEY trial and other trials that are ongoing now and that have been designed in the past few years, have undergone differences and tweaks and redesigns to really try to improve the benefits and what we can learn from each individual trial. For example, we now have, almost as a standard, if I look at it, many of these trials will have some crossover design where people in the placebo group at some point crossover into the active arm so that they do receive the benefit, albeit at a later time, of being exposed to the active drug. We attempt to stratify using different methods to maximize the ability to detect an effect. One method is to design inclusion-exclusion criteria so that the population is somewhat more homogeneous and we can decrease the size and the time of trials. One goal, which has not been fully achieved, would be to use what we know about subgroups and prognosis to identify individual subgroups within a trial that may have better or slower progression or better impact depending on mechanism. That is something that we're working on right now. We also are beginning to use biomarkers more and more within the trials, both as endpoints themselves, as well as ways of determining mechanisms and disease in the process of the trial. Next animation. So this is kind of my last couple of points. All right, so in the United States, there is a clear need kind of founded on all the people with the disease, about 30,000, and all the stakeholders. That's led to a very large number of preclinical studies and a fewer number of clinical trials. And the application itself nowadays is not simply to identify effective drugs. And certainly that is the main goal. But we're trying to design trials so that we can learn as much as possible from each individual trial. Final animation. And so, actually, no, there'll be one more after this. But the goal then is in the conduct of each individual trial, we have other integral components built in where we use the trial itself to study disease mechanisms, to determine the underlying pathophysiology using biomarkers and other studies of the trial participants. so that even in the course of the trial itself, we can identify other targets that we could use in the future for novel treatments, or even types of quality of life and other endpoints that we could use to directly improve patient outcomes. Final animation. And that's where the improved study design that we're working on, that's what we're trying to target. And so with that, I'll transition to Dr. Robbins.
Thank you so much, Dr. Sue. For anyone who has questions, please type them in the Q&A box. And I think we can move to the next slide. And thank you so much, Dr. Robinson, for joining us. I know we really value your partnership in research. I know you participate in many of our committees and have unique experiences as a pharma executive and also a person with personal experience with the disease. So I'll turn it over to you. Thank you.
Thank you, Sabrina. Just a brief introduction. I'm a psychosomatic medicine physician by training. I practiced for 10 years. And then I practiced in the pharmaceutical industry for 15 years and brought several drugs to market. Have had several phase 2. I've had a number of phase 2 drugs that looked amazing in phase 2 and failed phase 3 and so forth. So I've been through the new drug application process a number of times across a variety of different therapeutic areas. So let's go and talk about that. Next slide, please. So I'm going to go through really, and this is an incredibly, I just want to say it's an incredibly complex process involving many, many disciplines in a company. And that includes regulatory scientists, toxicologists, clinical pharmacology, animal preclinical experts. And then there's a lot in manufacturing, quality and control as well. And so there's a lot of people coming with their unique expertise onto a team to coordinate a new drug application submission. A new drug application submission, and I'm going to say new drug application submission, there's also a BLA, which is a biologic license application, and that's for drugs that are biologics like cell therapies, et cetera. I'm going to focus my time on new NDAs or new drug applications. When a company submits for a new drug application, it's really you're submitting now because you've got all the data, you've got your clinical talks, your safety, you've got your clinical trials done, you've got your manufacturing processes done and quality control done, and now you're saying to the FDA, I think we're ready to bring this treatment to market, and we think it has a potential benefit over risk. And so the purpose of so what the FDA is looking for is they're looking to see if a drug is both safe and effective, but also that the benefits outweigh the risks. And that we spend a lot of time on this section of the new drug application, particularly doing benefit risk evaluation. And benefit risk evaluation is also done in the context of the disease. We talk about that in ALS because ALS patients are willing to accept a little bit more risk. And so the benefit risk evaluation isn't something that's static. It's dynamic. It's influenced by disease characteristics. It's influenced by many factors. And it can be a little bit subjective as well. The second part of the new drug application that's really important is getting the product labeling correct. And product labeling, and we all have seen it, I mean, we get the little inserts of all the different safety and information around the product label. But that actually starts really early in development. In fact, we draft a product label well before phase 2, and we design trials with what we'd like to be able to show about data or about the product, working off what we call a draft launch label. And that is being updated as we get more data, as we get more learning, as we get more information in different sections throughout the drug development process. And near the end of the submission process, we really dive deep into talking about the product label. Does it accurately represent the benefit, risk, and the safety and efficacy of the drug? And thirdly, manufacturing methods and quality controls. And this shouldn't be emphasized. It really is an important aspect of a new drug application submission. And there's a lot of work that goes behind making sure an asset is ready for commercial manufacturing and that you've you've got good quality controls. The FDA does inspections at manufacturing sites. You've got good supply chain redundancy built in order to be able to, if you approve this drug, you want to be able to continue to supply it to patients. And so there's a lot of effort that goes into making sure manufacturing methods are good and quality controls are set up, and that we can continue to produce quality product to commercial supply. One thing to mention here, and I'm going to bring it because manufacturing changes throughout the course of a development life cycle. When we begin in phase 1, we're doing basic rudimentary manufacturing. We're getting it to a small number of patients. We then go to phase 2, and we're scaling up a little bit. But things may change in the process. The methods may change, manufacturing methods may change. So over the course of clinical development, you might not be using the exact same manufacturing methods in phase 1 as you are when you get to phase 3 and commercialization. Obviously, you want to document that. You want to make sure all your decisions are well documented and all the processing and controls are communicated with the FDA in a rational fashion. So these 3 aspects are really key to the new drug application. That's safety and effectiveness, labeling, and manufacturing and quality control. Next slide. So the main content, this is the content of what goes in a new drug application. And it's many, many, many thousands of pages of work. And before things were done electronically, you would literally send trucks of documents to the FDA. The main part, obviously, is clinical data. And this is the data and the results from human clinical trials. It's phase 1, 2, and 3 studies, as Dr. Sue pointed out. And it includes not only the safety information, but also the efficacy information. You have an important aspect of preclinical data. So Dr. Sue mentioned all the preclinical studies that can be done. And this is the results from not only animal studies, but also laboratory studies and safety and toxicology studies and pharmacology, how the drug is metabolized, where it goes in the body, how it is eliminated from the body. There's a number of different things that really the FDA wants to know, that we understand how the drug is working, where it's going, and how it's being used in the body. I mentioned earlier, sorry, I have COVID right now. So manufacturing information, this is a really big aspect of the submission, and it takes a lot of coordination to make sure that the clinical teams are also lining up with the manufacturing teams, and we're all giving the information and contributing the information to the submission at the same time. I talked about labeling. Again, we put together a draft label, what we'd like the product to be able to say it does, and the safety we think it's going to have. And we work from that right from the beginning. We adjust it accordingly as we learn about the information, and it culminates in FDA actually negotiating what goes in that label. And I'll talk a little bit more about that later. As I mentioned, there's a section of risk-benefit analysis. So we perform our own risk-benefit analysis. We present that to the FDA. And the FDA also does their own benefit-risk analysis when they review the information. Included in the submission is all the information about patents and all the legal considerations regarding generic competition. And there's some administrative information. And then included with this submission is also proposed marketing materials. Because what we want to do is we want to tell the FDA, okay, if you grant us this approval with this label that we're proposing, here's how we're going to communicate it to the public, both physicians and patients, about how the drug works, how efficacious it is, and potentially what are the safety concerns or use concerns. One thing to mention, I'm talking about new drug applications, is this is for, I'm assuming this is just a simple single drug. Remember, there are drugs that can also come with devices. So a drug can be given in a certain syringe device or a pump device. And all of those things also need to go into a submission. So sometimes it's both a drug submission as well as a device submission. And it gets ever more complicated as the things that you need to deliver around issuing a drug expands. Next slide, please. So I mentioned this before. I remember a time when we actually did submit via many trucks going to the FDA with the submission documents. It's now done under a common technical document. And there are 5 main modules to this common technical document. And the purpose of this was to harmonize submissions throughout the world. So for example, if I'm submitting for a drug in the US, what do I require the same information and the same things for Japan or China or South Korea or Europe? And so we tried to, it was tried to make sure we harmonize as much as possible so that we're not duplicating efforts of different needs across different countries. Some drugs are submitted, some submissions are done in the US first, some have been done in Canada first, Europe first. Japan tends to have about a 10-year drug timeline lag often, and different global regulatory bodies require a little bit different information, and that's where we get to regional differences. So then I talked about that because Module 1 of the Common Technical Document is the administrative information, and this is also where there's a regional differences. So you put the differences around, you talk about the differences for Europe or Japan in this section of the document. The next module is overviews and summaries. The next module is quality and pharmaceutical documentation, and then we have non-clinical reports and then clinical study reports. And just to give you an example, a single trial clinical study report of a phase 3 trial can be many hundreds of pages. And you're often talking about multiple phase 3 studies, multiple phase 2 studies, some phase 1 studies. There's a lot of information that's being compiled together in a submission. Next slide, please. So when we talk about submission, you've probably heard of different timelines, and there are different timelines for how long it takes the FDA to review an application. There's one caveat. If you're submitting a new drug application, for example, and it's an antidepressant, it's got a similar mechanism of action to stuff that's already on the market, your standard review time is going to be 10 months. A priority review time is going to be 6 months. I'll talk about that. If you're, however, submitting a new drug application, and this is a new molecular entity, meaning there's nothing that the FDA has ever reviewed, there's nothing on the market that has this mechanism of action, it's a brand new molecular entity, the typical review time is 12 months. And for a priority review, it's 8 months. So overall, that's why you hear there's different timelines for a new drug application. It's based on whether the drug is similar to something that's already on the market or something that's absolutely brand new, never been seen, never been heard of before. And that's where those differences in timeline come around. So it's generally we're working on a 10 or a 12-month review clock. The course of the review, and I want to, in item number 4, the review conduct, during this time, you've got your whole team. The FDA is going back and forth with questions. They're questioning the team. Very often, you're pulling your statisticians together. You're running new analyses for the FDA based on questions they've submitted to you. You've got questions on potentially on pharmacokinetics, on potential toxicology, on potential safety. And so you're answering as a team, you're pulling together all these functions from your team and you're writing the responses to the FDA. So this is a back and forth process during this time that often requires a lot of team effort. That's also in addition to what you've already submitted to the FDA. So during this time, the teams are very busy. Placed on top of that, if in the development discussions, you anticipate having an advisory committee, and that's the conduct advisory committee around month 8 there. So in addition to talking back and forth with the FDA on various questions it may have, teams are also preparing for a potential advisory committee and trying to identify the key issues that will be discussed during that advisory committee. Next slide, please. You've also probably heard of various different designations of approval or of reviews. The first two, a fast track designation is done or is granted to a sponsor at the time of IND submission or pre-IND review. A fast track is in order to qualify for a fast track, you have to have a serious condition and potential to fill an unmet need. And preclinical data is the data that's acceptable to define getting a fast track designation. Breakthrough therapy is also done at the time of IND submission, or can be done also at an end of phase 2 meeting. And this again is for a serious condition. And now you have not only preclinical data, but you must have clinical data that is suggesting a substantial improvement over existing therapies. These two are not mutually exclusive. So you can both have a fast track and a breakthrough designation in a submission. The next two designations you've probably heard of now is one is accelerated approval. Accelerated approval, there's no formal process in terms of timeline when that gets granted, but it has to be a serious condition. It has to have a meaningful advantage over existing therapies. And the third criteria is you have to, what you're saying is, I'm going to approve this drug faster than the regular timelines of review and approval. And I'm going to do it before I have all the clinical data in, but I have some surrogate marker or surrogate measure that correlates to clinical effectiveness that is going to say this drug likely works. And so it's typically a biomarker. It could be a blood measure. It could be any number of things, but there's a surrogate endpoint of clinical efficacy. And that's granted for, and that you see a lot of this in oncology. This was first done in the days of HIV drug development. And that's when accelerated approvals came to light. And then the last designation is priority review. And priority review is done at the time of the submission of the new drug application. Again, it has to be a serious condition, and there has to be significant improvement over existing therapies. You'll hear about priority review vouchers. This is often a strategy that's taken, meaning these can be purchased as well. And so when some companies acquire other companies, sometimes they're acquiring them for the priority review voucher in order to get a faster review cycle. So there's a little bit of politics in the priority review designation in terms of vouchers being purchased and business development opportunities done to capitalize on getting a priority review. Next slide, please. So there's one or two responses that you're going to get. You're either going to get a complete response letter or you're going to get an approval. A complete response letter is the official communication from the FDA when a drug or a therapy is not going to be approved. And that letter will include information about what is deficient in the application, what data might be deficient. It could be clinical data. It could be labeling that you're not agreeing on. It could be manufacturing issues and deficiencies. It could be any number of reasons. It doesn't need to just be about efficacy and safety. You might have an efficacious drug that's safe, but you have some serious manufacturing issues that are raising concern and ability to provide quality product at a commercial supply. So and there have been also times where FDA and sponsors get caught up in what goes into the label. Label negotiations are a particularly back and forth time. You get on a call with the FDA and you negotiate what language is going to go into the label. That becomes important when you're thinking about marketing the drug to physicians and patients, because what goes in the label is basically what you can say proactively to physicians and patients. So we really, really pay attention to and spend a lot of time on label negotiations. And we strategically plan what we're going to fight for in the label and what we might be willing to give up. And sometimes you get to a fork in the road and you can't agree, and then the FDA issues a complete response letter. There's three options when you get a complete response letter. You can resubmit the application addressing the deficiencies. You can completely withdraw the application, or you can request a meeting. These are typically called Type C meetings, when you request a meeting to discuss what the issues are and see if there's a path forward to an approval. Next slide, please. The approval letter, we all get excited, and typically all the approval letters that I've got have been after 5 p.m. on a Friday. So it tends to be a thing with the FDA. But it's simply, you'll get a letter saying it's deemed safe and effective for its intended use. You'll be given the product label that you've all negotiated and agreed to. The FDA bases this based on all the information that was in the submission as stated before. Next slide, please. So the approval process involves all the aspects of what we've talked about: clinical trials, manufacturing data, product labeling, and in addition to the approval, you can often have an approval with conditions. So that means I'm going to approve your drug, safe and effective. You're going to be able to market it and provide it to patients. But also, we need you to do another study in, say, geriatrics, or you have pediatric commitments. And so there can sometimes be conditions for approval. It could come with risk mitigation management, or a REMS commitment. It can come with commitments to do a registry of patients prescribed the drug. So there's various different things that the FDA can say, I'm going to approve it, but I also need you to do this. And so that can have an impact on resources on the team, and making sure that the team is resourced to satisfy the conditions of approval. Next slide. So that's a really brief overview of a very complex process, and we're happy to take questions. Dr. Su.
Thank you so much. I mean, this was fantastic. What an overview. And I have to say, I particularly appreciate the fact that you shared some parts of the process that are not widely talked about, or can be very confusing, just if somebody just, reads, on the news or website. So really great to have this specific insight. So we have a few questions, and I know we're having a little bit of a longer webinar than usual, but that's so important. So hopefully people can stay because the questions are also very interesting. And maybe we can have you back. I don't want to to ask for more. And thank you also, Dr. Robinson, for being here despite COVID. So the first question is actually for Dr. Su, and it's about what drives ALS progression and what biomarkers could be useful in addition to neurofilaments.
So that's actually a pretty complex two-part question. The different factors, we recognize several, and we know that there must be several that we don't understand. The last cartoon that we had up, actually that streetlight bias cartoon, that kind of highlights that to a certain degree, right? So it's that classic conundrum where it's supposed to be a drunk person who's searching for their car keys under a street lamp in a dark night. Policeman comes over and says, "What are you doing? You're looking for your keys. Are you sure that the keys are over here under the street light?" And he says, "Absolutely not. I lost it somewhere else." And so, "Why are you looking here?" "Because this is where it's bright. This is where I can actually see." Right? So there's definitely processes and techniques that aren't available right now that probably would help. We're really trying to use existing techniques to uncover these. In terms of what factors are underlying disease progression, I think clearly there is what we know about the degeneration of motor neurons themselves, and then we can certainly pick up on that degeneration using neurofilament. Other important biomarkers kind of look at different stages and processes in the disease. I mean, there's a lot of biomarkers for both the mechanisms themselves. The biomarkers for endpoints aren't as well elucidated. So aside from neurofilament as a blood biomarker, and then the various clinical endpoints, the actual functional rating scale, the breathing, and things like that, specific serum, CSF biomarkers for endpoints, they're not quite clear. But biomarkers, what we're exploring include, for example, we know that upper motor neuron predominant ALS proceeds a little bit slower than lower motor neuron predominant ALS. We're trying to see, are there ways that we can tease apart why that is? We know that specifically respiratory is extremely important in ALS. So all the trials look at respiratory very carefully. Certain compounds are thought to impact respiratory and bulbar a little bit more. So in those trials, these monitoring mechanisms for speech, for talking are biomarkers that we look at as well. But admittedly, it's something that's well beyond what I can expertly talk about in a short amount of time.
Yeah, and then we have a fun question, so to speak, which maybe for Dr. Robinson, how are drugs given their name? When does it happen?
Yeah.
And why do drugs have different names in different countries?
Often, the drug starts out with an acronym or usually a name relative to a company. So ABBV 308, for example. After that, it gets a chemical name, and the chemical name is the generic name. So duloxetine is Cymbalta, for example. So there's the generic name, and that's basically based on chemical structure, et cetera. The brand name, which is what you're referring to in terms of the commercial name, it actually is a team of people that deal with this, and there's two things we want to consider. You want to be able to find a brand name. It's hard to that has some indication of what the drug might be for or do. So, for example, launched a drug that we called Duodopa. It was for Parkinson's disease. It's on dopamine and duo. It acts both on two different types of. Anyway, so you try to pick a name that has some aspect of what the drug does or what it's being treated for, etc. But at the same time, it can't be a name that explicitly claims some efficacy claim. And so there's a real nuance there, and a real subtle nuance. The other thing is it gets run through a whole number of databases. It can't be too similar to a family name. For example, I was working on an oncology asset, and we had this drug name. We thought it was great, and there was some Italian last name that was fairly common that was the same name, and so we couldn't use it. Legal said we couldn't use that. Then there are languages, so you have to think about that as well. And so some words in different languages may have different connotations. I remember getting a name, and I would just put www.name.com, and up comes an illicit site. And so in a different language. So there's a few nuances. You have a team of people that's doing that. You want to communicate some information about the product and what it does, and it can't be a name, it can't be a company, it can't be a trademark anywhere else. And so that's a key aspect to the branding that goes, how do we choose a name?
Thank you. Yeah, and the reason I thought this is a fun question is because I believe there are companies that specialize in this, checking that the name of the drug is not offensive in any language or things like that. And there's actually a science and an art to it, which is kind of interesting. Dr. Su, why do so many drugs pass phase 2 but do not make it through phase 3?
Yeah, right. So, the goals of phase 2 are essentially to allow any drug that has a reasonable chance of efficacy, a small signal for efficacy, to pass, and also to allow somewhat safe drugs to pass. These phase 2 trials are smaller, and unfortunately, when you have a smaller, shorter trial, the statistics behind it means that while it may look effective, once you get into a much larger, more powered trial, it may prove to actually not to be effective. I think if you go into the basic science realm and ask bench scientists, they will say that a large proportion, mostly they will say the majority of their experiments end up not even close to achieving what they want. And so, the fact that phase 2 trials in general, overall, are achieving about 33%, and then phase 3 are 25%. If you actually translate that to the bench, and that actually is a much more better track record than most of your typical preclinical or basic trials. That's not to say that this is an excuse or a way of justifying the lack of success, but the way that rigorous scientific approval and determination of efficacy is such that we really want to make sure that the phase 3 definitive trials don't allow drugs that aren't effective to pass. And I think it's because of that high standard that so many ALS drugs in general don't pass.
Let me add just one thing to that. I've had two programs that I've been responsible for that had absolutely outstanding phase 2 data that failed phase 3. And there could be many reasons for that. But one of the reasons we had was on one of them, the phase 2 was largely conducted at a few sites in Russia, had outstanding data. And then we went to global. And as you sometimes, your global footprint and what countries you include can vary from phase 2 to phase 3. And that can have impacts that you're not really able to measure, but are significant.
Yeah, and one question for you, Dr. Robinson. You refer to advisory committees at the FDA, and so there's a question about sort of how does that work? I mean, is there a specific ALS advisory committee that the FDA consults with, or can you tell us more about that?
The advisory committee, so there's a neuroscience advisory committee, and there's a standard number of potential reviewers for that. And then there's community reviewers that can come in. The FDA puts these together in advance. ALS advocates can apply and can put their name in the hat with the FDA as do I want to be considered for something like this. So there is a standard neuroscience advisory committee. Know that though, not all the members on an advisory committee for ALS are actually ALS experts, and that's one of the challenges with the advisory committee. And that's why that's so important for the community section and presentations for other ALS experts to come and present their opinion on it, because not everybody on the committee is going to be an ALS expert, and that does create some issues and can create some issues. You try to manage that by bringing in the community members and ALS experts to the community section portion of that advisory committee.
Yeah, and if I can add to that, I think that it's a little bit by design as well, because you want people to be people who can, the participants, the panelists, have to be able to judge the science and the data, but they're generally excluded if they have what's considered a perceived conflict of interest, which doesn't mean that they actually have a conflict, but if they are consulting or working closely in that space, there is a perceived, a conflict with their role. And therefore, oftentimes, actually, people are not experts in that particular disease area, although they're experts in general in neuroscience, but not in that particular area.
For example, as Sabrina said, I was asked by the FDA to participate as a community reviewer on an ALS advisory committee, and because I had worked in pharma, I couldn't do it. So that was a conflict of interest. They're very, very strict about their conflict of interest.
Yeah, even if they are sort of not directly related, but even if there is something that could be perceived. So that's something to keep in mind. So actually, there's a question for you, Dr. Robinson, because you have this experience in the pharmaceutical industry on the pharma side. Can you explain why the few people with ALS can get on trials or EAPs, and why is there a kind of a restriction on who can participate in research?
As you know, ALS is a very heterogeneous disorder, meaning there's a wide variety of patients with ALS. When you're trying to do a clinical experiment, what you want to try to do is you want to try to study patients that are most similar to each other. And when you're studying ALS, which is a progressive neurological condition, you want to be able to measure any treatment difference over a reasonable period of time. That's primarily why we focus on faster progressing patients and try to make the patients the most similar. That's done with inclusion-exclusion criteria. The downfall of that is that roughly only 10% of the ALS patient population overall can actually qualify for a clinical trial. One of the most restricting exclusion factors is whether you're either 24 or 36 months from symptom onset. So if you've had symptom onset over 24 months or past 24 months, you won't qualify for most clinical trials. We're doing that in order to be able to detect a treatment difference as reliably and as quickly as possible. So it's just by a factor, just by the nature of the disease being a progressive neurodegenerative disease where there's a wide range of different patient progression rates. We focus on fast progressors, which actually limits a lot of the inclusion to about 10% of the overall population. EAPs, there are two main reasons why EAPs may not get done or are challenging. One is manufacturing, and two is resources and cost. It costs a fair amount to execute an expanded access program, and many of these companies don't have a commercial product or a revenue stream to draw on if they're small biotech companies. So that cost can be a real impediment. That's where Act for ALS was hopefully made some big difference. The other thing is manufacturing. Manufacturing changes over the course of the development timeline. And very often we're challenged just to meet product supply requirements for a phase 2 trial. So adding more patients for EAP can really strain the manufacturing system. So it really depends on the complexity of the manufacturing, on the number of sites being used, on the supply chain for creating the product. So manufacturing can be an impediment to doing EAPs. And the third reason that EAPs may not get done is a myth, in that it gets communicated that there's a regulatory risk. So if you're going to do an EAP, you're using it in patients that are not in the clinical trials that could develop or could raise a new safety signal that you didn't already anticipate, and that may lead to a delay in review. Of all the EAPs in total history done, only one has ever had that happen. And so it's really a myth. It's not a huge regulatory risk to do an EAP. And then the next question is, well, when do you do an EAP? Can you do it in phase 1, 2, 3? Most EAPs get done in either parallel to phase 3 or after phase 3 submission. There are some EAPs that get done in phase 2. We see some of that here, but majority of EAPs get done phase 3 or after submission.
Yeah, and thank you. And you also addressed the question about when to do an EAP, and just wanted to put a plug on our EAP webinar series. If you have more questions for the EAP, we'll take them there as well. I know we're almost at the hour, and I think the reason people keep sending us questions is because this is such a fantastic and unique webinar. We've been wanting to do this for a while, and hopefully we can bring both of you back because this is so interesting. Maybe for both of you, a couple of questions that I'm going to summarize in one, and I'm going to ask you to for one last comment from both of you on this point. There's a couple of questions about what other endpoints could be used, what other outcome measures could be used. So for example, you spoke about neurofilaments. Are there other biomarkers or surrogate endpoints? Or we know that the FDA has been using, the ALS-FRSR as an outcome. Are there other outcomes that could be used for clinical significance? So maybe we can conclude with thoughts on both, because I think those are actually great questions to think about how the drug development process is going to change over the next few years. Who wants to start?
Dr. Sue?
Yeah.
I'll start. I'm passionate about this topic. I think outcome measures are our number one risk factor for finding drugs in ALS. In fact, in that we need to improve our sensitivity and ability to measure progression in a heterogeneous group of patients. That's already a challenge. So I think there's actually work being done on this. There's a task force that's working on this. There was a recent harmonization of the standard operating procedure for the ALS FRSR. Just as a side note, there were two different SOPs, US and Europe, and they weren't exactly the same, and they didn't score items exactly the same. And it was only recently that the SOP is harmonized into one exact same SOP. So there are a lot of nuances there, but I think I hold a lot of promise in this area. I think outcome measures is one of our biggest areas where we need to improve. That's my personal opinion.
I think there's a lot of debate in the field about the best outcome measure. The ALS-FRS right now is a gold standard in all of our clinical trials. We do recognize that one of the big problems with the FRS is 1 point is not the same for any two individuals, right? So a trial where a drug has a person in the active arm decline by 4 points, if that person was, doing very well, fours in all the domains, and each of those points were individual points in domains that don't really impact quality of life, for example, versus another person who declined 3 points, but they were all in respiratory, and the person was in 3 and is now 0 and requires ventilation, non-invasive ventilation 24/7. There's efforts to develop other scales that are what we call RASH, harmonized. So this is a method of having scales where each point on the scale is truly linear, and we can analyze and compare directly in a more apples to apples way. I will say that despite all of the types of different methods that we're trying to develop for outcomes, in the end, usually what we look at are the most objectively meaningful clinical endpoints for people, right? So we could always look at dynamometry and look at the amount of grip strength or something else like that. But 5 pounds for one person, when the person is really truly full strength and can lift 50 or 100 pounds, is very different than a person who has minimal strength and goes from bending their elbow at 6 pounds now to only 1 pound. Right. Whereas the FRS is something where we try to look at things like objective, important things to people. Are they able to eat on their own, write on their own? So kind of a long-winded answer, but the different outcome measures themselves, we're trying to improve to focus on not only the clinical side of things, but also maximizing quality of life.
And this is where we need more consensus.
Yeah.
If you look at trials done in the last, say, 10 years, you've got ALS FRSS, you got the CAFs, you've got Munichs, you've got MITOS, you've got a whole variety of different primary outcome measures being used. Kind of indication to me that we need more consensus on what's best to use.
Yeah, definitely. On that note, thank you so much. Thank you both. Obviously, we had great turnout and lots of great questions, and thank you so much for staying longer and for sharing your expertise. And hopefully we can have you back soon. Thank you. Thank you both.
Thank you, guys.
Thank you. Bye.